morning, afternoon, or evening to you wherever you are in the world. My name is Michael Gore, and I'm editor of the Plant Phenome Journal. The Plant Phenome Journal is the only plant phenotyping journal supported and run by a scientific society. All TPPJ articles are open access and receive targeted promotion within the plant phenotyping community through Wiley and Tri-Societies. Today, we have a wonderful webinar planned for you. And this Access Journal webinar today will be given by Dr. Jared Crane. Dr. Crane is a research assistant professor in the Department of Plant Pathology at Kansas State University. He has a focus on implementing genomics-assisted breeding and plant breeding programs. His research has included both high-throughput genotyping and phenotyping, and how these methods can be integrated into plant breeding to increase the rate of genetic gain. The title of his webinar today is Field-Based Single Plant Phenotyping for Wheat Breeding. So spaced single wheat plants were evaluated using high-throughput phenotyping methods to extend high-throughput phenotyping to the earliest stages of plant breeding. And this is an application of affordable high-throughput phenotyping that could provide plant breeders with exponentially more data to make selection decisions throughout the plant breeding process, leading to increased genetic gains. With that, Jared, please take it away.
Yes, thank you, Dr. Gore, for the introduction, and thank you, everyone, for joining online with whatever your time zone and day time may be. And I'm really excited to present some of the work that I've been doing in high throughput phenotyping, looking at wheat breeding. And I would like to start. This is the main research article that I'll be presenting data from, and I would definitely like to acknowledge my co-authors, Zhu Wang, Byron Evers, and Jesse Poland. I've got the opportunity to share this work with you, but without their contributions, this work wouldn't be possible. It's definitely a team effort, so I really want to acknowledge them and appreciate their efforts throughout. So the webinar today, I just want to introduce kind of the motivation for plant phenotyping, and then why we went to this single plant phenotyping effort, and at the end kind of looking at future applications where I see this going in the future. So just to set the stage for high throughput phenotyping and why we would want to introduce that into a breeding program, this is a diagram that shows the Kansas wheat breeding program, and you can think of the program as a large funnel that starts out with bulk populations. These are segregating plants. As those advance in generations, then you go to individual plant short rows down to 3,000, finally entering about 300 in yield trials, and continuing on down the funnel and evaluation until one or two new varieties are released. And thinking about, so that's the breeding funnel flow of germplasm. If we think about the flow or the quantity of information, it runs the opposite direction, starting, maybe at this individual plant short rows, maybe you can genotype that. 3,000 plants is still going to be fairly expensive. And then as this moves through the breeding pipeline, the types of test, everything from grain characteristics, flower quality, that are both going to be more labor and time intensive, as well as monetary cost, those tests go up. So this is just an example of the Kansas program. In the same thought, another picture of a funnel that for a large wheat breeding program, I believe this was based on the CIMMYT program in Mexico. Again, follows much of the same path. We start with lots of F2 segregating plants. This gets whittled down throughout the funnel until the F10 generation, where a couple varieties are evaluated, and 1 or 2 are recommended as varieties. So within this breeding funnel, when we look at the rate of genetic gain, the one formula that I try to keep in mind, the breeder's equation, we know that our rate of genetic gain is a function of the selection intensity, selection accuracy, having genetic variance within that population, and the time that it takes to complete that cycle. So high throughput phenotyping really provides an opportunity to quantitatively measure some of these traits that are correlated to final traits of a variety that we're interested in, such as grain yield. This is just two pictures, 2 graphs of high throughput phenotyping traits. On the left-hand side, this is NDVI, so normalized difference vegetation index, plotted against the grain yield in grams for small plots. And NDVI usually has a very strong relationship, positive relationship. The higher that NDVI value is, some ways you can think about the greenness of the plant. The more green and robust that plant is, most likely the higher that plant yield is going to be. And we see that trend through this data. In the same way, another high throughput phenotyping trait is canopy temperature. Within wheat breeding, we see usually a negative correlation where the cooler that plant canopy temperature is, which usually relates to evapotranspiration and maintaining a cool temperature with sufficient water supplies, the higher our grain yield is throughout. So these are just two examples of traits that could be collected in a high throughput manner for hundreds or thousands, even tens of thousands of plots. And some of my first work in high throughput phenotyping, or HTP for short, in wheat breeding, was looking at, when we think of that funnel, the preliminary yield trials. So these are plants that have already been inbred. They've already been scaled up to seed multiplication to get to a small plot, 2 meter by 6 meter plot that can be measured as a yield trial. And this is just a diagram of canopy temperature. In total, there's about 3,900 plots, with each small segment representing 1 plot here. So this was some of my first work. Again, we were looking in this preliminary yield trial already quite a ways down in the breeding funnel. So this work really was motivated kind of by the question of, can we increase the rate of genetic gain if we can move that high throughput phenotyping earlier into the breeding program? And for this case, we tried to go as far up the breeding funnel as possible, essentially to thinking F2 segregating plants. And those are going to be all single plants. So that's where the idea and motivation for single plant phenotyping really came from. So to test this idea out in practice, we put out 2 different trials, one called the single plant association mapping, or the SPAM trial, and then we'll see in a second the association mapping, or just the AM panel. And this used 340 genotypes. Many of those lines, about 280, were in the hard winter wheat association mapping panel. So they've been used in a variety of studies, not just this particular experiment. And for single plants, we started a single seed in the greenhouse to ensure that we only had 1 seed, 1 plant. And then later on, about 3 weeks later, those single plants were transplanted into the field on a spaced grid. And this picture just shows everywhere there's a pink flag or blue marking would be where a single plant was getting transplanted into the field. And we did this over 2 growing seasons, 2018-19 and 2019-20. With the main change in the second year, we went from a 50 centimeter grid spacing up to a 70 centimeter grid spacing, just to ease some of the post-processing challenges that we found in year number one. So these plants were grown in the field throughout the growing season. We used high throughput phenotyping with uncrewed aerial vehicles, so UAVs, that were collecting RGB imagery and near infrared imagery so we could calculate our normalized difference vegetation index, or NDVI. In addition to the HTP data, we also collected a lot of hand measured data, including plant height, spike length, spikelets per spike, and the number of tillers per plant. So this data was all collected on the single plants. And then right beside the single plants, we had the association mapping trial. And these were the exact same genotypes, but they were planted in two replicates in yield plots that were 2.4 by 1.4 meters. And these plots were managed for optimal yield. And the same way with the single plants, we collected NDVI with UAVs and then hand measured the same traits that were collected on the single plants, with the number of tillers being per unit area instead of a single plant. So this is just an orthomosaic of those two experiments and showing how they were grown side by side. So this is the 2020 SPAM plants. Each one of these small green areas is basically an overhead view of a single plant. And you can see there's areas where we do lose some of these single plants. If the plant dies, then it's going to be an open spot in the field. And then right beside this, within 10 meters is the full yield trial of the same genotypes that are two replicates. And looking down over these AM panel plants, we can see each plot, and then areas where this looks smudged, it would be lodging throughout the field. So we experienced a range of everything, and these lines, there were the older, taller varieties that were prone to lodging as well. So the last bit, once these images were collected through the UAV, is data processing. Zhu Wang was the main person handling the data processing, but I really want to point out that there's a lot of steps from collecting the data on board the UAV, all the way from checking the data, making sure there's ground control points, moving to an orthomosaic image just like we saw. Once we get to that point, then we can take our plot boundaries that are the full plots or the single plants and extract any trait of interest. For our example, we'll be looking at NDVI, which is near infrared and red imagery. And once you extract those pixels per plot, then you can take the mean or the mode, whichever measure of central tendency you would like to have the plot value. So the question then is really, how did this look? So this is a correlation between those single plants and full plot yields between 2018 and 19. And on the top row, this is grain yield of those full plots. The bottom row is grain yield of the single plants. So there was a negative correlation between single plant yield and full plot yield. And then the following columns are all correlation between NDVI and grain yield. So on the bottom row, this is the NDVI of the single plant correlated to the grain yield of the single plant. And there's a positive correlation, a strong positive correlation throughout. On the top row, this is the correlation of the single plants to that full plot grain yield. So we see really strong negative correlations. Year number one, we think, aha, this is great. we just go look for plants that have a very negative correlation. We put this out year number 2, and we see a much different picture. This year, there were no significant correlation between the single plant or full plot grain yield. No correlation between the single plant NDVI and full plot grain yield. However, we do continue to see a strong correlation between the single plant NDVI and single plant yield. So just looking at this, then we looked at across years trying to decompose the variance components for these traits that were measured, grain yield, and then handheld traits like plant height, spikelets per spike. And I want to zoom in on spikelets per spike, where one of the variance components due to the genotype variance between both AM and single plant is very high. We have low residual error variance. And with that, we looked in a GWAS just to see if we could find the same peaks. And on the top Manhattan plot, this is the full plots, and we see a significant peak on chromosome 7A. In the single plants, same trait with the same year, there wasn't quite any markers above a FDR level, but the 2 most significant markers were the exact same as the full plot. So this really gives us some evidence that what we're seeing, the genetic control in the full plots, we can also see in the single plants. And then just looking at that variance again, when we see, look at other traits like grain yield, we have much higher residual error variance, very little variance partition to the genotype. And when we tried doing a GWAS, unsurprisingly, we didn't find any strong marker hits in those on those traits. So I'd just like to conclude with kind of some future applications where I see opportunity for single plant phenotyping. And from this study, which I would call just kind of the first proof of concept, the tip of the iceberg, we know that we're going to have large G/E interactions. As informatic methods improve, we could see a way to use as a single plants that aren't going to take much up space up. We could plant some designated checks that we know are really good, plus a check that is going to be lower performing, as a way to identify in season which plants we may want to carry forward. And ideally, this will give us multiple rounds of selection, before plants are hitting preliminary yield trials. So hopefully we can increase that rate of genetic gain. And of course, enhancing informatic methods are essential. When I think about high throughput phenotyping, it's a combination of software, hardware, biology, and informatics. And this area is continually improving, everything from using commercial software to developing pipelines to handle the data. And it's very much a team effort. This is just an article by Dr. Shi at Texas A&M. And I just want to highlight, they looked at multiple crops, but they had several objective teams to implement this high throughput phenotyping. It quickly becomes an effort that it takes more than one person or one lab to implement this in a real-time fashion. So the last few thoughts that I'd like to leave everyone with, and not to discourage anyone, but to make sure that everyone's aware of what it takes to implement this, is there is a large lead time from developing the idea and to the implementation and actually using these pipelines during the breeding season. In particular, thinking about, can the data be processed, analyzed, and selected in season? If one day of flights takes a week to process the images, then there's no reason in collecting images in the field more often than once a week, because the timeline, you wouldn't be able to process it and wouldn't get any extra benefit for those measurements. And, finally, our breeding programs, prepared for cheap, large amounts of phenotyping data, and how does that look throughout the program. And I would like to just acknowledge this was funded by the USDA NEFA Eager Award, and lots of lab members transplanting, measuring field. There was a team of 15 to 20 people in the field collecting that data. So it does take a team effort. And with that, I really appreciate your attention, and I will turn it back over to Dr. Gore as the moderator.
Great. Thank you so much for your presentation today, Jared. I'll give you a virtual clap there for you. Okay, so there are some questions, and I will ask the first question if that's okay, just to help get people to think about some questions to submit to the question and answer chat. Okay, so what are some constraints to implementing high-throughput phenotyping single plant analysis into current plant breeding pipelines?
Yeah, so constraints of implementing some of the HTP and single plant, I think it could even scale to full plot. It really is pipeline development. When we look at UAV imagery, being able to process that in a real time or very quick fashion, the plant breeding cycle, if it takes you 2 months to process the data, but you've already harvested the plants, it's very hard to make in-season decisions. And I think that's something that, it has greatly improved in the last 5 years, and I think there's continuous improvement on making sure that we can do that, but still an area that is very active in research and needed as plant breeders start looking at these methods.
Great. Thank you so much for that response. That's helpful. Okay, so 2 questions on the transplanting side that we'll address. So I can just read them from the question and answer window here for you, Jared. Okay. So what was the method for the transplanting of single plants from the greenhouse to the field?
Yeah, so the single plants were started in a small plug, just with potting soil. And then it was a kind of a biodegradable, I'm sorry, I don't have the exact name, but essentially the entire plug, which the roots could grow through, was planted just in the field. Okay. And a related question is wheat transplanting a usual practice? Why not direct seeding and thinning? Okay, yeah, very, very good question there. And yes, it's definitely not a usual practice. And we wrestled with this question before we started, do we just go to the field and plant a single seed? But we went with transplanting just to make sure that there were no volunteer plants. The field was tilled before transplanting, and also to make sure that we didn't have any extra tillers if there were multiple seeds and they were close together looking like a single plant with tillers. We wanted to be sure we just had a single seed. Okay. And then there's a continuance of the question on the single plants. The data is negatively correlated with the plot yield. How is this method then applicable? Yeah. So at least in 1 year, it was negatively correlated. The second year, we didn't see correlation. I think that's really environmental. We had 2, the first year with negative correlation was, pretty optimal growing condition. The second year was a much more challenging, really droughty throughout that booting, tillering growth in the spring that we would like to see. And I do think like the negative correlation really comes back to idiotype breeding. Donald, a 1960s paper, suggests that for wheat, you want a weakly competitive idiotype that doesn't produce tons and tons of tillers, because these plants that we see high NDVI, one thing that was really surprising is you could have tillers on a single plant, space planted, in the 50s and 60 tillers per plant. So you had these really vegetative plants. They did produce a lot, that produced more seed than a plant that didn't. But if we think in full plot, we don't want those plants competing to produce all those tillers. We'd like a weakly competitive plant that produces a few tillers and seeds on those tillers. I think using that again is a little bit challenging because we know we're going to have G by E. I do wonder if we could plant, when we start looking at space planting, having multiple checks, a very elite variety, an older variety that we know is not as good. And then we could basically correlate where on the spectrum the relationship NDVI falls between what we know should really perform well versus one that doesn't. I think we really just started, again, kind of proof of concept, tip of the iceberg. There's a lot of work that could still be done in this area.
Okay, so there are many questions, and I will try to get through as many as I can until I'm told to stop. Okay. Did you make any corrections for heading date or maturity?
We did use heading date as a covariate in the model.
Okay.
Just heading date, not flowering time.
Okay. Was the germplasm genetically the same and isogenic between the full plot and single plant plots?
Yes, the germplasm should be the same. It would have came all from the same seed source. In fact, we had one seed source that was used both years for the single plants and plots. And that was the reason we went with the association mapping panel, is because it gave us an opportunity to plant that single plant that was genetically the same year one and year two. I mean, we do envision like moving to an F2 population where there is a true segregating plants. But for testing, we wanted to make the most reliable duplicating opportunity possible.
Okay, great. Thank you. It's quite interesting that you found overlapping GWAS hits, which suggests high reliability, but do you have concerns about not capturing random effects by using single plants? And I'm assuming this is the field effect variation. Yeah.
Yeah. And I'm sorry, concerns about not capturing field.
Well, it says random effects by using single plants. I'm assuming random effects is just the variability that's happening in the field. Yeah.
That is a good question. With the single plants, to capture in an effort to kind of try to mitigate that, we did go to higher number of replicates. But there still could be, at a single plant, you're much more likely to either place that plant in a really optimal or bad spot, where a full yield trial is going to be a little more spread out. So there is, a very, very good thought and question. And, we did our best. I'm not saying we were perfect at accounting for some of those effects. Okay.
So unless Emily tells me to stop, I'll just keep going past 10:30. Okay, do you recommend I'm sorry, I'm just trying to read all these questions. Do you recommend phenomic evaluation rather than genomic evaluation, considering the cost involved in the two systems?
That could be a very viable path forward, because that would, if you went to strictly phenomic evaluation, you would reduce your genotyping cost, and the UAV imagery is relatively affordable. There is an upfront cost with UAV computing power, but then you could easily use that same equipment year in, year out. I don't have any, we didn't go and look at that particular question in this data, but I would say, again, very, very open question, very ripe area for research in plant breeding.
Okay. So the next question, does the 1 week to process data from 1 day apply to both association mapping and single plant association mapping?
Yeah, so the question was about the timeline of processing data, and I should clarify, when I was given that was just an example. Zhu Wang actually did all the data processing. I would really have to check with him on timeline, but I know stitching, making those orthomosaics can be a time-consuming process. So I just want to, again, put that out for people that even though you can go collect lots of UAV data to try to analyze it in season and make decisions, there's more than just the collection that you should be concerned about.
Okay, great. Regarding the negative correlation between yield and canopy temperature, we see this across crop species. What do you think is the physiological explanation for the relationship?
So there's been a lot of work on this, and a lot of work at CIMMYT with Matthew Reynolds' group. I don't know that there's a full one thing, but a lot of data supports, possibly higher root biomass, longer rooting depth, getting to that water, open stomates, so allowing transpiration to happen instead of, closing the stomates and keeping the water in the plant that's still going to keep that cooler. So there's several viable hypotheses, and I think maybe all of those have an impact, but I don't know that there's one confirmed specific thing in wheat breeding for sure.
Okay, so I'll just kind of re-ask a similar question that has been asked before, but there's still like continual related questions to this that have come, even though you've somewhat addressed this topic before. So if you bear with me, Jared, I'll ask again. Since the correlation between the yield from single plant and plots is low, what is the advantage of the single plant phenotyping for yield breeding? What is your next step for single plant phenotyping?
Yeah, so we know with our data across 2 years that correlation between single plant and full plot yield can definitely be variable. 1 year we had fairly significant correlations. The next year, absolutely nothing. I think in application, there is some opportunity to maybe look at yield, but also with high throughput phenotyping, and we didn't dissect or go this route. But a lot of that early inbreeding is selection for disease resistance or some other, visual trait. So I think there's opportunity even to look at RGB imagery, some of the, advanced AI, artificial intelligence methods, to where you could go scan a field, run the data through the computer, and it could identify plants that you're going to want to carry forward or plants that you want to cull multiple times through the year, rather than just the breeder passing through one time. And this is an ideal plant, we're going to carry it forward. This is a plant that we need to delete.
Okay, great. All right, so I'm going to the other chat window now. There's a whole other set of questions there. And thank you for being so accommodating, Jared, to answer all these questions. Okay, take a breath. Okay.
Yes, and I just wanted to point out as well, we don't really have a time limit. We just try and keep it to 30 minutes. So if anyone wants to stick around and hear the answers to these questions, that's fine.
Yeah, I hope you all can hang out, because I'm very eager to hear all these answers as well. And I'm sure Jared is happy to stay for an extended period of time.
I will stay and cover as much as we can.
Awesome. Okay, let's just do this then and have fun. Have you tried other vegetation indices as well?
Just within the paper, we just looked at NDVI, and we didn't delve into that again. But if you're collecting this data, you're so close to being able to implement all sorts of different vegetative indices Yeah, I'll just say for ease, we just looked at NDVI and said that was enough. But it's definitely quite doable with the technology now, that once you get the base implemented, multiple vegetation indices, canopy temperature, which would be thermal, everything from like LIDAR, could all be on the table to analyze through some of these same pipelines and methods.
Okay, yeah, that makes total sense to me. Okay, so what software did you use for analyzing single plant phenotyping? And I guess this could be a few different software, so take your time. Yeah.
Yes. So I'm going to assume this means a lot toward the image data, and the both AM and the single plants making the orthomosaic, that was handled through AgiSoft. So that is a commercial software that you pay for a license. And then so that made the orthomosaic. This processing pipeline, there is a pipeline developed through Python code implementing the AgiSoft toolbox or package within that. So it's as less user operable. I mean, it's punch the button and let the pipeline run on all the data, rather than individually inputting images and stitching and making orthomosaics. So the pipeline is essential for moving the quantity of data through efficiently.
Okay, great. Hopefully that answered their question. Okay. So the weed management on 70 centimeter spacing could be challenging. Are there strategies you recommend to make weed control feasible in an operating breeding program?
We did, and Byron Evers was, he was really the plot manager, and he did the phenotyping as well. But they were all managed basically for optimal yield. Both years there was herbicide applied. Have to look, it's in the manuscript specifically, but to keep weeds down. And I think, trying to start with clean fields helps as well.
Absolutely. So speaking of Byron, he has a question for you here. Have you compared multivariate trait heritability between single plant versus full plot?
We have not, or I have not with that. I mean, I do have for individual traits, the heritability, and they do both in the full plots and single plants. The heritabilities look, they're moderate to high. So we have, confidence in the data we're getting. But again, there's so many emerging areas and really cool analysis that could be performed. It's just where do you stop and say we've completed all we can based on the manpower. So.
Sure, that makes total sense. Okay, I think you already answered this one question. Plants in the field look crowded. How did you separate single plant from other plants?
Yeah, so that was the first year we were at 50 centimeter grid, and the second year that was the reason to go into that wider 70 centimeter grid spacing. Using the grid, we were able to take advantage of a grid to identify where those plants were, and then it was essentially just a spatial area around that center grid point was taken for a single plant. Now, we do know there was a little bit of overlap, but I think that overlap, as this technology advances, I think we'll start dealing with that overlap, because it's not probably feasible for a breeding program to single plant on 70 centimeters, but maybe you can space plant your F2s enough to where you can pinpoint down to this is the small area, one or two plants in this area, and a breeder can go in and choose the the plant that they want out of an identified area.
Okay. Well, we still have 100 or more of your closest friends still on this Zoom, and there are at least 2 more questions to be answered. And this next question is also related to single plants, and this is just a comment. We can't undermine the efforts of the conventional breeders of the past, some are still, who made visual single plant selections for whatever traits and for all the tremendous progress that was made.
Mm-hmm. Yeah, I would just add with that. I mean, yes, the idea of, call it early generation single plant phenotyping selection, it's an older idea. We're really just putting new tools into it. There's a really good review by Fisher et al. 2018 that looks through a lot of efforts that were conducted through the 60s, 70s, 80s, I think, even 90s at single plants, and traits that were measured that are correlated. The challenging thing is many of those traits were very time and labor intensive that would not scale to a breeding program, whereas we think some of the UAV work would be able to scale to tens or hundreds of thousands of plants at a time.
Okay, great. Okay, next question. With the rate at which computer power increases, is computational time requirements really a matter of concern relative to real-time data processing?
Yeah, that's a good question. I don't. It's probably going to come down to program specific, location specific. If you're at an R1 institution where you have a high performance computer with thousands of nodes and CPUs, it's probably not an issue. Now, if you're trying to implement this in maybe a developing country or somewhere where your biggest computer is a large workstation, it could be an effort or something to keep in mind that would slow you down. Okay,
great. So I don't see any more questions. And if I didn't ask your question to Jared, please repost it because I likely just missed it in all of the flurry of posts. But I'm going to ask one maybe question in closing. Assume there aren't any other questions to be asked. Where do you see the application of field phenotyping in the future? What areas of high throughput field phenotyping need to be improved so that they are routine application in plant breeding programs?
Yeah, so I really see this again as something that in the future it's going to take more work, more effort, but that plant breeding programs can affordably implement and start looking at inbreeding material F3, F4 for hopefully both positive and negative selection. But to get there, computing power or ability, pipelines. I know, and even in the last 5 or 6 years, the ability to orthomosaic photos and extract data, may both probably in terms of ease of use and developing those pipelines has greatly increased. So as that continues to get more user-friendly, even the pipelines we used, require a fair level of computational skills. It's definitely not a GUI user interface where you can just click around with the mouse. So trying to get those tools to where they're easily used by a broad audience is going to be essential in getting that high throughput phenotyping out there. And on the analysis side, again, we started with single spaced plants, with this idea of push phenotyping to the far edge of the breeding pipeline as we could, or the breeding funnel. But in practice, we're probably going to need to be able to handle plants overlapping and try to work with multiple plants, even though where we can't separate them completely, but at least have the idea and geographic location if you have GPS data where those 1 or 2 plants are. So that's kind of where I see this going in the future. Lots of opportunity, I think an area ripe for more research as well.
Okay. There is one question that was reposted. I think you had maybe addressed this one actually already in your slides and in the Q&A, but I'll ask it again. What is the correlation between single plant phenotype and multiple plants phenotyping of the same genotypes?
What is the correlation between, with this question, is it possibly leading toward the phenotype of NDVI between those? I mean, for grain yield, we know that single plants usually have a positive relationship between NDVI and grain yield. The correlation between that single plant grain yield and full plot is variable. One year we found very nice negative correlations. The next year, you know, absolutely no correlation. And I would have all the data. I actually don't know that I've looked at it for, you know, an NDVI of one plot versus the NDVI value of that same plant. But the data would actually be there.
Okay. Cool. All right, Emily, I think that's everything. I guess if there are no more questions, then let's thank Dr. Jared Crane one more time.
Thank you. And again, I would
fun.
like to acknowledge the co-authors. I mean, without them, this definitely isn't possible. So I appreciate all their efforts as well.
Yeah, we just want to thank everyone for joining us today, and thank you to Dr. Crane for sharing your research and answering so many questions. As a reminder, the recording will be made available and sent to all attendees soon. Thank you again, and have a good day, everyone.
Thanks a lot.
Goodbye.
Thank you.
Goodbye.